Hello guys, my name is Dr. Varun and you are watching chlorophyll classes only on YouTube. Well, in this video, we are covering our 12th class topic, that is the next after what we're going to be studying from the translation topic. Now from translation topic, we have studied that how with the help of RNA, we are able to make the proteins. Now, once the protein is being made, what will happen after it? Its regulation is needed. That we will come later on. First, let's just complete one important section of the protein synthesis translation. That is that you know that the RNA is what? It is a different types of RNA there, in which mRNA is the one which provides it's a long chain kind of a thing in which the codons are present. What is codons? Basically, it is what? It is the code for the protein synthesis, right? That's why we call it codons. Now, what is codon? I said that codons is nothing but the triplet of the nitrogenous base. What does that means? It means that this one triplet means 3 nitrogenous base comes together and forms one codon. Let's just see that. That is, our today's topic is genetic codon. So the genetic codon is nothing but what? It is the nitrogenous sequence or nitrogenous base sequence present on the mRNA, which will help in what? Deciding which protein, what will the protein code. Okay, so which amino acids will align in the protein synthesis, okay, which determines the amino acid sequence for the polypeptide. What is polypeptide? Protein, okay, polypeptide. Now, if I say that okay, tell me just a little bit more about it so that I can understand better. So that is what that one codon is made up of 3 nitrogenous bases. 3 nitrogenous bases. Like for example, if this is your RNA, okay, which RNA? mRNA. Okay, this is the one. It is 5' to 3', and it has sequence. Okay, what is the sequence? I'm writing it up. A U G C C A U U U C A U A G C U A C A. Like this, it is the what? mRNA. Okay, this is the mRNA. Now, if I will ask that how many codons are present on it, so codons are what? One codon is equal to 3 nitrogenous base. Okay, so there will be what? 1, 2, 3, 4, 5, 6, 7. 7 such codons are present on it. So what was my question? My question was, how many codons present on given mRNA? So we have calculated it. What? The answer is what? That is 7. Okay, we understood this thing, that what is codon and what is basic thing we have understood. But how we know that in one codon there are 3 nitrogenous bases only coming? Why not one only? One nitrogenous base should be one codon, 1 or 2 or 3 or 4. How come this 3 thing came? Here comes the permutation, here comes the probability factor. Okay, probability and permutation. So what is that? Those students who don't have math, It's okay, don't worry, I will let you know. Probability, you know na, that we are able to put the number of chances that what can happen with it. Something we know about that thing that is coming up over here. Don't confuse that. There is the no questions which is coming over it. We just putting up the thing so that in academically you can write an essential reason you can understand. Okay, so if we see that, so here we come certain discoveries. So what discoveries we gonna be talking? Number one discovery is that how do we know that the codon is triplet? Okay, so name by name we're gonna be doing. So there was a person named George Gammon. So what he said? He proposed that it will be what? It will be a, what we say that that the genetic codons would be triplet. Okay, proposed that genetic codon will be triplet. Now the major question is how it is triplet. So for this, first thing that George Gamow knows that is that there are total 20 amino acids that is needed in our body for the protein synthesis. This fact was clear to him. So let's see that how the different 20 amino acids will be there. Just imagine that this is the one thing and on this different amino acids are present. Let's take an example. So imagine this is the first thing. If 1, 2, 3, 4, 5, 6, 7. So we know that if one nitrogenous base correspond to one amino acid only, means one thing is making one codon, then only 4 amino acids would be made, because the work should be what? We know that one thing will make only one kind of a thing. This also remember it, fact. So means what? This A, U, G, C. This will make one, this will make second type, this will make third type, this will make 4 types. So only 4 types of amino acids possible. Okay. Which is not the case. We know that there are 20 types of amino acids present. So it must be joining up together, means those 20 such in the different proportion joining up in the final chain formation, which we have done it in the last video. I hope you are able to understand this. Okay. So, okay, if this is not the case, then second option it taken is what? That let's take that if it's not 20, it's not 4, then can we take it 2, 2? Okay. So it's take what? It takes this option that 2 is acting like what? 2 is acting for codon. Now over here what option is there? Like for example, if 2 nitrogenous base correspond to 1 amino acid, then probability of number of amino acids becomes 16. How is that becomes? So if you see that whatever is the 2 raised to power n, okay, this is a tough one. Let's not write in the permutation form. Let's just write it like, for example, this is A. A can make bond with A only. A can make bond AU, AC, AG. All 4 are making difference. Let's see over here. Then C can be there. CU, CA can't be made. Okay, so that will be what? CG. Okay, because C have already made, so that will make the same bond only. Then comes the GG and GU. This thing is already made. This thing is already made. Can you see this? This thing is already with the G. And the last option would be what? That would be UU. Okay, so if you see this, how many such are being made? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 only. Okay, so 10 only combinations coming. But how many such things are present. How many amino acids are present? Amino acids are 20. Means what? Then this also don't make the sense. What make the sense now? The sense is being made by, or you can put it in the Punnett square also. This is really tough if you're not able to understand this thing. Simple thing is that, of course, it will be 16 only. I made little less. Like A, U, G, A, U, sorry, G, C, G, C, C. So these all options would be there. So means only 16 amino acids would be there. But we know that we have what? 20 amino acids. So finally, triplet will make all 20, or correspond to all, will correspond to all 20 amino acids. But the question is that how many such will a triplet makes? Now the triplet will make 64 such codons. So triplet probability of permutation will make 64 codons, which are unique. These 64 codons make only 20 types of amino acids. Okay, so if you see that how the whole thing will look like, let me just show you. See over here. So this is it. Can you see that there is one letter nitrogenous space, another nitrogenous space, and third nitrogenous space, and they are making what? Triplets. And these triplets are making what? These are making the corresponding to this amino acid. So like triple U, which correspond towards what? Phenylalanine. UUC will correspond to what? Phenylalanine. Means two different codons are making corresponding to same amino acid only. Did you understand this thing? So it says that more than one codon will be corresponding to one amino acid, but each codon is having the unique one, and that's only. It won't go to do anything else. This is important property also, later we will come to know that. So we understand this thing. So this is what? This is the first nitrogenous base. This is second nitrogenous base, and these are the third nitrogenous base. Okay, this thing we understood. Here what you have to remember, that there are certain nitrogenous base, what they are making you should remember. First of all, there is what? This one, UUU, it will make phenylalanine. CCC, it will make proline. AAA, lysine. And GGG, glycine. Okay, so these four you will remember, always you have to remember. So this will make phenylalanine, this will make proline, means not make, whenever I'm saying make, it means correspond. Okay, don't get confused. This will make lysine, which is a basic amino acid and the part of histone proteins, and this will make glycine. Okay, let me know in the comment section whether the glycine or the aniline, which is the simplest amino acids, and what is the reason for calling it simplest one also. If you know that, write it in the comment section. Now this thing we remember. After then, this one more thing you have to remember, that out of 64, only 61 of them are actually code for an amino acid or correspond to an amino acid. Okay, out of 64 codons, only 61 corresponds to amino acid. So what about the rest 3? 3 of rest are stop codon, or it is also called as nonsense codon, which do not code for anything and it acts like what? Termination point, if you remember the last topic. Okay, to nonsense codon which do not code any amino acid or correspond to any amino acid. What are they? UAA, UAG and UGA. Okay, so here are those stop codons which you have to remember. Wherever that will come, the process will halt. So over here I am giving you one question on this basis. Okay, try to do. This is the amino acid. The question is, how many amino acid will be present on polypeptide chain formed by the given mRNA. So you have to count what? Number of codons between the start and the end codon. Start codon is always AUG, okay, and the stop codon could be those three. So you have to just identify those. Okay, let's just count it. This is the start codon. So from here it will start, keep on here it will stop. So means this much only will be participating. So let's just mark it up. So this will be what? That 1 2 3 4 5 6 7, and this is what? Stop codon. So this would be what? The number of amino acids. Okay, so means 7. 7 is the answer. So this type of questions are much expected in your academic examination, means your board examination, as well as in the NEET examination as well. So this you have to remember to identify this thing between this. So whenever something is given, if AUG should be the initiating point and the ending point would be the stop codon, it could be either of the three. Okay, before it only will be the part of the polypeptide formation. Okay, so this we have understood that thing. Okay, now he told that this is it, but who will give the experimental verification for it? For that there is a person from the Indian origin, that is Har Govind Khurana. This person actually told that this codon triplet is correct, but he is not himself able to do all the things. There was multiple research which has taken place which he utilized and give it this thing. So what are the two things? Two different experiments that has been taken place in the discoveries that took place. Number one discovery, the second discovery which is important for us, that is the sequence. Okay, triplet has been given, people are still finding it, Har Gobind Khurana did it, but how? The two things which we have to remember, we will be doing it. So the first discovery that we need to know is that by the Severer-Okhwa, this person find an enzyme which is important for the phosphorylation, which means what? To add the nucleotides together. So that this enzyme is able to do. Okay, so it helps in what? It find the enzyme that is polynucleotide phosphorylase, which help in polymerization of RNA into defined sequence which we want, means which nitrogenous base we want to put, okay, into defined sequence without any template. Means normally what? RNA will be made on the template will only be making what, which will be what? Code. Okay, we don't want that, we want our own nucleotides to be put forward. Okay, so this is what has been done without any template. Okay, very good thing you have find it out. Then comes the Marshall-Nirenberg. Marshall-Nirenberg's. What this person did? This person is able to polymerize a RNA in the cell-free area. Without the requirement of any cell, he can make the RNA, because normally what this whole thing is taking place inside the cell only, right? So he is able to make cell-free synthesis of RNA molecule. Cell-free synthesis of, not the RNA, of protein molecule. Okay, I don't know what happened. Protein molecule. Okay, so this thing we understood that they too did it. Now with this information, can we see that, like for example, we can take this enzyme, we can make the template, and after that outside the cell only we will ask them, okay, make the protein out of it now. So these two things which Hargobind Khurana used. Okay, so fourth is the Hargobind Khurana. Experimentally proved that codon is triplet. So if codon is triplet, how we will come to know that? We will remember the fact that 3 will make 1 amino acid. Okay, next will make next amino acid. Let's see that. So what he did, he cell-free synthesized the first he synthesized a RNA molecule. So which type of RNA molecule? Two different. One was homopolymer, and another one is copolymer. Okay, homopolymer means he took the same type of amino acid. Okay, so sorry, not amino, he takes the same type of nitrogenous base. Okay, so means RNA consists of only one type of nitrogenous base. Let's take an example. So this is the RNA. Okay, let me just make it with the red only, so that you will always remember that the red thing is RNA, which we have made in our notes. Okay, so this is 5' to 3', and all of this has what? U. Example, okay, U means uracil only has been added to it. Simultaneously he did what? He took another one where he was doing what? Copolymer. So in copolymer, what is happening? In copolymer, RNA has two different types of nitrogenous bases, one after another. Okay, which means what? Like this time he take U and A, so it becomes UA. It becomes UA, like this. So first thing, he's used the enzyme from the experiment of Silver and able to make this according to his own wish. Now the Marshall's experiment, he can synthesize this also. So he did what? He let it what? Protein synthesis. He undergo what? Translation. So the next step was translation of this process. Now after translation, what happened? You will get what? You will get the amino acids accordingly. So this chain is being made. Okay, here also the chain is being made. When he analyzed, you know that triple U will respond to what? triple U will respond to this thing. Can you see this? That is phenylalanine. So here what happened, this whole chain, when he analyzed, there were all what 100% phenylalanine is present as the amino acid only. So as it analyzed, it has 100% phenylalanine. So he understood that a single will be making what? Same thing only. Understood also, na? Phenylalanine. But if he has put one after other, so what happened? This become one, this become another. So what is repeating? UAU or AUA. So this is repeating. Now UAU correspond to what? See, UAU correspond to tyrosine. So what happened? There is tyrosine is being formed as the first, like that has been put it on the first place, okay, and AUA will form what? See this, A, where is U? This AUA will form isoleucine, okay, so it will form what? Isoleucine. This is what? Isoleucine. Now he understood what? When he analyzed it, analysis shows two different amino acids, okay, that is confirming what? That is confirming it's a triplet. That's it. Because if it would be two, okay, what would be happen? If it would be two, like someone will say that no sir, why not be taking two? See over here, two will be what? UA. It should be similar result like the first one, homopolymer. But the copolymer things that are coming up is different. It's not same. So we here come to know that this is your what? This is your triplet, not the doublet or the singlet. I hope this thing is understood. Okay, if not, then let me know that. Okay, in the comment section, I will help you out much better. Now let's come to the next thing. This is what we understood, that how it's happening, discoveries and everything, and we confirm that okay, this is triplet. It will have certain characteristics. Let's just try to learn that. Okay, so this is the properties, which is very important from the all point of view. Okay, so this should be that in your markings, that don't leave this, leave everything but don't leave this. Okay, so next is properties of genetic code or genetic codon. Now, so number one property, which is the easiest, and we all know that the nitrogenous bases are in the form of triplets. So codon is what? It is triplet. Okay, so codon is triplet. Second point about this is that it is non-ambiguous. Non-ambiguous means, ambiguous word means very ambitious. Like for example, I am a very ambitious person. I was a dental graduate. I can do dentistry. But I went to teaching. I also like creative field, so went into that field also. I like the interiors. I went to that also. So I am doing what? I'm exploring a wide variety of sense. So this is called as what? It is called as ambiguous. I'm very ambiguous person. But these codons are non-ambiguous. Means one codon will code for only one amino acids, and that is also we have seen in that box also. Okay, so genetic codon is non-ambiguous. That is, one codon corresponds to only one specific amino acid. Exception is there. What exception is there? Normally your start codon is AUG, but sometimes what happens, GUG comes on the start position and start coding for the corresponding towards the methionine, formanyl methionine. That's the problem. Okay, so there is what GUG normally codes for valine. Okay, GUG normally, or better way, GUG is, this is the only codon, is ambiguous, very ambitious. It's like me, who want to teach also, who want to do dentistry also, who wants to do other things also. Is ambiguous codon as normally codes for valine, but if it comes at the start position, comes at start or initiating position instead of AUG, if GUG is present, initiating position, it codes for methionine. Methionine only becomes what? The start of the polypeptide chain. Okay, then the second one, the third one. Third one is that degeneracy of code, which is what just coming from this only fact. We know that same type of amino acids are coded by two different codons. Okay, why? Because it is a degeneracy of code, means code is not very unique. Okay, if someone is not doing, other person will be coding for the amino acid in that place. Okay, so degeneracy of code. Degeneracy of code means that one amino acid is coded by, same amino acid is coded by more than one codon. Example, UUU codes phenylalanine, UUA also codes for phenylalanine. So if you see over here, so UUC. So if you see that over here, this is also phenylalanine, this is also phenylalanine. UUA and UUG is leucine. Okay, this also C thing is also for the leucine. So many of the codons are coding the similar or corresponding to the similar type of amino acids. This is what, it is not unique. So it is what? Degeneracy. Okay, so only, okay, one fact here pe, which we will remember over here, that is only methionine, AUG and UGG, that is UGG is what? This tryptophan. Okay, UGG, tryptophan codes by one only. Okay, that is the only thing. This you have to remember. So these are the facts, things that you have to remember about it. So degeneracy, understood now, that similar amino acids can be coded by the different codons. Codon can code only one. Okay, that is, first one is degeneracy, latter one is the thing which I spoke, is the non-ambiguous nature. Okay, next thing is, when we are reading it in the form of what? Continuous, without any comma, without any pause. Okay, so continuously it will be written up. Means what? If you are working and making the polypeptide, you will read continuously. Even if any one nucleotide goes away, we won't say that it is gone. We will read the next one and accordingly correspond, and everything will change, and that's where the mutation comes, and that's how the sickle cell anemia comes. Okay, let's just go through it further. The fourth point is that genetic codon is commaless and is read in continuous manner, continuous fashion, without any punctuation. Next is this genetic codon is universal, means all over the organisms present will have this thing only, means they will be coding for that way only, and it will be triplet only. It will be coding like UUU in me, in bacteria, in plants, in any organism which is present will code for phenylalanine. Now, if it is, there must be some exception, because everywhere exception coming, here also exception comes. So in mitochondria and certain protista, it is not there. That's why it is said that they are so different from us. Okay, so the last thing, next point is genetic codes are universal. That is, one codon code for one particular amino acid in whole living world. In whole living world. But exceptions are there. Exceptions, that is mitochondria and certain protists, belongs to protozoan. That is it. Next is the codon is, or codon are non-overlapping and do not non-overlapping, that is, they are independent of each other. It's not like that this will work, or like that. Independent of each other, they are working. Okay, that is, each codon is independent and do not overlaps with the next. That's the properties. After this, there are two terminologies that we will just write the headings and remember from this video. Okay, that is what one is wobble hypothesis. What is wobble hypothesis? Wobble hypothesis says that normally the first, like in a codon, there are three nitrogenous base, but the first two is being read, the last one is not even important. So most of the cases, the first two is read, and what amino acid will come, it already know. Okay, so the third one is not even read that much. Not always there, but most of the cases. Okay, so it is known as, just the heading we will be writing. Okay, the third nitrogenous base in a codon is generally does not change the expression of codon to a particular amino acid, so does not make a much difference. Okay, after this is one gene, one enzyme hypothesis. Now we always used to say that one gene is responsible for a character. So what happened? There was two person, Tepetum and Biddle. They told what? That one gene will make only one type of polypeptide. You know that gene will make the mRNA will make the proteins. So it says that one gene will make only one enzyme. Later on, of course, Baltimore and some other people, Jacobson came and they modified this thing and says that one gene means one polypeptide that will be making to express its function. Understood? So it says they just told about one gene, one enzyme experiment, later on modified as one polypeptide, proteins that is being formed. Understood? Now the next thing is the gene regulation. How can we regulate the genes? So in eukaryotes and prokaryotes, different functions are there. So what are there? why the gene regulation is needed. Like for example, if there is any function which needs to be changed or enhanced, or maybe certain things which has to be done, and that is what where the gene regulations takes place, which is responsible for what? Protein synthesis. So next is regulation of protein synthesis. Means at what step we can check it to stop it or see that whether it has taken place or not. So at what step regulation of gene synthesis can be done? So first of all, we're going to see in eukaryotes. In eukaryotes. So first of all, first step would be what? To make the protein, you had to go through the process from DNA to RNA, RNA to protein. So if we stop the first process, that is what? DNA to RNA formation. That if we can do what? We can regulate at the formation of transcription, that we stop the transcription process. This is very important for the later on in our understanding for the treatment of certain diseases. Okay, so the first step is, at multiple steps we can do it. Okay, so first step that we can take it is the transcriptional level. Transcription level, we will say that okay, stop, we don't want to make this protein. Okay, so at transcriptional level we can do it. If we are not able to stop it at, what will be the next? RNA has formed. Now the RNA will be forming what? Inside the nucleus, because DNA will be present what? Inside the nucleus in the eukaryote. Now RNA next has to what? It has to make the proteins, but proteins won't form in the nucleus. It will form into what? Into the cytoplasm. So from nucleus it has to come out. Now this come out process is what? This process is the translocation. So we say that at translocation or during transfer of RNA from nucleus to cytoplasm at this time. But the RNA that is being formed, it is hnRNA. Means it is what? It is not completely what we say. If you have studied the transcription, if not, go back, see it again. Then it is not completely mature enough. So what we can do? So they can actually process it to make post-transcription level. If we stop that process, splicing, editing or capping, tailing, then also it won't be able to stable enough and won't be able to survive. It will be degenerate after a certain period of time. So we can do it at post-transcriptional levels. We can make it a change. Or even if we are not able to make it, then during the keeping a check over the translation process, means from RNA to the protein formation, we can stop at the translation process also. Translation level. So these 4 times we can do it. But now think that what about the prokaryotes? Okay, so in prokaryotes, we can only do at 2 levels. One is transcription level and one is translation level. Why not the in between 2 things? Let's try to discuss. First point is transcription level, and second is translation level. Now the question is, why not at other 2 levels? Other 2 levels same as eukaryote. Now let's try to understand the first point. Second point which is not present. Translocation level. Translocation means what? From nucleus the things goes out. But do they have nucleus? Do they have any boundary? Do they have any cage? No. Where in the prokaryotes, if you remember the prokaryotic cell, then what happened? Prokaryotic cell has nucleus present in the cytoplasmic area itself. No covering is present over there. If no covering is there, means whatever the RNA will be formed, it will be directly going into the cytoplasm only. First point. So we will say that first would be what? As no nuclear membrane present after transcription, RNA formed is directly in surrounding of cytoplasm. Okay, first question done. What about the second point? Why this thing is not there? So beta, if you would have gone through the transcription process, you know that there is a post-transcription thing which I told that it only and only occurs in the eukaryotic cells. Because in eukaryotic cells, it is what? It is the enhanced versions, because eukaryotes are what? They are monocistronic RNAs, means only one protein forms, so they do that whole process and make it more stable by adding the things. But here the such things is not there. They don't undergo the post-transcription level itself. So second is prokaryotes do not undergo do not undergo post-transcription process. So if they don't have process, how we can manage it? Okay, do not undergo post-transcription process. So what else? Is there any other way of regulation in prokaryotes? Yes, it is, and that is called as operon, which will be our topic for the next video. I hope you are liking these videos. If you are liking this video, give it a like, share it with your friends, because that will help me to motivate me a lot. Do subscribe me and put the bell icon, and I'm putting videos on every day so that your syllabus could be completed on time. Well, that's not all the things that we are doing. As I told you that we are coming with something really good. So this is the announcement. You might have even seen one of the ad in between where I was talking about it. Here I'm telling you once again that you can talk about your doubts, everything. Even your parents can do it. If any parents is watching it, they can also come up and ask us the doubts, whatever they have regarding the prices, details and everything. We are waiting for you on the Instagram's DM with your queries and everything. Well, that's all for this video, and thank you so much.